Raja How. This is Ruchi Brian James S. Lagitnay presenting to you a research about bacteriocin regulation gene and its role on the PCC's bacteriocin production. Bacteriocins are antimicrobial peptides produced by microorganisms acting as DNAs or RNAs to its target bacteria. PCC is a bacterial plant pathogen causing soft rot diseases to many plant species. This bacteria is also capable of producing bacteriocins. In our laboratory, we have isolated carosine S1, carosine S2, and carosine S3, all are types of low molecular weight bacteriocins. In this study, it focuses about BRG gene. This plays a pivotal role in the control of gene expression in bacteria by stabilizing sRNAs and facilitating their pairing with multiple target mRNAs. It is then hypothesized that this BRG gene can influence the bacteriocin production, regulates the other bacteriocin regulatory genes, and the type 3 secretion system. Thus, this gene was cloned and was knocked out. And this was electroporated into the host cell PCC89H12 for homologous recombination. In vivo studies revealed that under the influence of ultraviolet irradiation as inducer, we have observed higher or bigger zone of inhibition with that of the wild-type strain, while smaller zone of inhibition was observed with delta BRG mutant. This is due to knocking out the BRG no longer possess the low molecular weight bacteriocin but still possess the high molecular weight bacteriocin. Under the influence of glucose induction at a varying concentration, it was found out in these in vivo studies that glucose cannot induce bacteriocin production, as you can see from the different patterns of zone of inhibitions being presented. In the in vitro analysis, it was found out that at the transcriptional level, delta BRG mutant can no longer transcribe caro S2K and caro S3K. These genes are responsible for the expression of carosine S2 and carosine S3 respectively. Moreover, the FlyC, which is a part of the type 3 secretion system gene, is also not being transcribed in the delta BRG knockout mutant. It is then proposed in this study that under the influence of ultraviolet irradiation, the BRG gene can facilitate in the translation of the bacteriocin regulatory genes CRP and DGC, which are responsible for synthesizing carosine. And it also acts as a chaperone in ATPase complex, thereby facilitating in the secretion of the bacteriocin via the type 3 secretion system. It is concluded that BRG knockout mutant was unable to produce the low molecular weight bacteriocin, and carosine production of the BRG knockout mutant is inducible by UV irradiation but not by glucose addition. And BRG is potentially required for carosine externalization through the type 3 secretion system.